Dámy a pánové, já vás srdečně vítám v nejvyšším purkrabství na Pražském Hradě u další ze série přednášek Věda na Hradě, kterou pořádá Správa Pražského hradu společně s Ústavem organické chemie a biochemie Akademie věd České republiky, Karlovou univerzitou a také děkujeme za podporu nadačnímu fondu IOCB Tech, díky kterému se vlastně tyto přednášky můžou konat. A dneska mám tu čest přivítat vzácného hosta, který přiletěl až ze San Franciska, pana Tomáše Cihláře.
Děkuji.
A který nám dnes pohovoří o tom, co jsme se za 40 let naučili o HIV. A já bych poprosila profesora Konvalinku, kdyby byl tak laskav, a pana Tomáše Cihláře uvedl. Děkuji.
Děkuji moc, paní Wolf. Dámy a pánové, já mám to veliké potěšení a čest představit doktora Tomáše Cihláře. Já to říkám pokaždý, že mám potěšení a čest, ale tentokrát to opravdu myslím vážně.
Já už ti začínám věřit.
Tomáš Cihlář je český chemik, americký chemik českého původu, který se narodil podobně jako řada dalších vynikajících českých vědců v Chomutově v severních Čechách, a kde taky vystudoval gymnázium. Potom vystudoval VŠHT, ale my se na něj nezlobíme, protože pak pokračoval, pochopil, kde se dělá ta správná věda, a pokračoval na Ústavu organické chemie a biochemie Akademie věd, kde udělal PhD. Mimochodem nikoli jako školitel pod Antonínem Holým, jak se o něm říká. Pracoval v týmu Antonína Holího, ale jeho školitelem byl doktor Votruba, což je mimochodem velmi významný člen Holého týmu, který spolu s Ivanem Rosenbergem, Hankou Dvořákovou a řadou dalších významně přispěl k tomu epochálnímu úspěchu těch protivirových léků. Tam pracoval na biochemii a virologii virostatik vyvinutých v týmu Antonína Holého. Potom odjel na stáž k firmě Gilead do Foster City u San Francisca v Kalifornii a tam mu to docela šlo, takže se postupně stal až viceprezidentem firmy Gilead Sciences. Tomášovi zásluhy o virologii a vývoj virostatik jsou mimořádné a určitě se mi je nepodaří shrnout. Já bych řekl, že nejenom pracoval velmi úspěšně na studiu mechanismu účinku tenofoviru a dalších virostatik na vývoji rezistence vůči němu, ale později byl šéfem vývoje dalšího preparátu, původně vyvíjeného proti hepatitidě C, jestli se to správně pamatuju, později úspěšně klinicky prověřenému proti viru Ebola, který nakonec se stal prvním klinicky prověřeným a schváleným lékem proti COVID-19 pod jménem Remdesivir. Vedle toho a spolu s tím, a o tom bude, jak doufám, Tomáš vyprávět, vedl výzkum ve vývoji dalších virostatik proti HIV, které mají tak neuvěřitelnou účinnost a tak dlouhodobou účinnost, že dramatickým způsobem mění způsob, jakým vlastně v tuto chvíli zvládáme tu chorobu a mohou vést potenciálně až k jejímu úplnému vyléčení. Tomáš Cihlář je vynikající vědec, ale takových jsme už tady pár viděli a v tom vlastně výjimečný není, protože těch výjimečných vědců je tady víc. Co mně připadá fascinující na něm je, že se takhle vynikající vědec může stát současně významným představitelem velké nadnárodní farmaceutické firmy. A Gilead je jednou z největších farmaceutických firm na světě dneska. A co je ještě zajímavější, aspoň pro mě, je, že se významným viceprezidentem velké farmaceutické firmy může stát člověk, který je laskavý, slušný, čestný, korektní a milý. Pro mě to je strašně důležitá a dobrá zpráva o stavu dnešního světa. Těch dobrých zpráv nemáme dost, takže si ji pojďme užít. Tomáši, těším se na tvoji přednášku.
Dobrý večer, tak doufám, že mě všichni slyšíte. Já jsem hrozně rád, že tady mám tu příležitost na Hradě trochu povídat o HIV a chtěl bych říct pár věcí. V první řadě opravdu chci poděkovat panu profesoru Konvalinkovi za několik věcí, nejenom za vyčerpávající a možná i trochu nerealistický portrét mě samotného a ale také za to, že to vlastně byla jeho idea, abych tady měl tu možnost přednášet, které si moc vážím a za velmi pěkné uvedení. A pak samozřejmě hrozně děkuji organizátorům a sponzorům té akce, o kterých se už tady mluvilo. Takže dnes strávíme zhruba asi 50 minut z HIV a ponoříme se do té rané historie a budeme se snažit i trošku přemýšlet nad tím, kam celý ten boj těch 4 desítek let nás dovedl, co jsme se naučili a jak to můžeme v budoucnu nějakým způsobem uplatnit i v obraně proti budoucím virovým pandemiím a epidemiím, které určitě na nás se budou dál valit. A ještě poslední poznámku, kterou bych chtěl říct, já se musím velmi omluvit, že ta přednáška bude v angličtině. Já jsem byl dost rozpolcený, zvlášť tady na Hradě, to by bylo dobré vlastenecké gesto, ale pak z praktických důvodů jsem se rozhodl, že tady vás nebudu trápit 2 hodiny koktavou češtinou, protože musím připustit, že moje poslední přednáška v českém jazyce byla před 30 lety při obhajově mé aspirantury v Ústavu organické chemie a biochemie. A od té doby ten můj český odborný slovník se vytrácí. Takže, ale můžu vám slíbit, že snad se pokusím otázky a odpovědi, aby jsme měli v češtině, ale tu hlavní přednášku teda budu mít anglicky. Takže let's switch the languages. Let's start with HIV. So HIV is a human virus that infects the cells that on the surface express the protein that's called CD4. Unlike many other viruses, it actually can infect only 2 types of the cells, CD4 T-cell lymphocytes and macrophages. And it is a relatively small virus, but it's very infectious. It's RNA virus. It has about 9 kb of RNA genetic material, and most of the proteins that are expressed in the infected cells are in the form of polyprotein that needs to be cleaved. As every other virus, the only function of HIV is to get into the cells and multiply itself, make many copies that then spread across the same cells and cause the disease. And so here is just a very simplified scheme of the replication cycle of HIV. It starts with entry, reverse transcription, which is basically a process that changes the RNA genome into the DNA, and that's why the HIV is also called retrovirus, as many other retroviruses. That's the hallmark of the retroviruses. Then when the DNA gets into the nucleus, it gets permanently integrated into the human genome. And that's the big issue with HIV, because the copy stays there. The CD4 cells are long-lived. That's our part of our adaptive human immune memory, and these cells live for years, and that's the main reason why HIV now these days cannot be cured. It can be only treated or prevented. And then in the end, when the proteins are expressed from the integrated profile DNA, the virus gets assembled in many copies. Now, what is important is there are few enzymes that are specific for the virus. They don't exist in human body. Virus brings them into the infected cells. And these have been historically parts of the replication cycle and also good targets for the antiviral therapeutics. Now, how HIV causes the disease? The transmission, it's so-called blood-borne pathogen. The main transmission route is sexual, but especially during the early stages of HIV epidemic, a lot of people have been infected by transfusion and blood products, which nowadays doesn't happen because every bag of the blood is actually screened. Every donor is screened for HIV and many other viral pathogens. IV drug users, when they share the contaminated needles, they also get infected. But I think what is probably the most drastic realization is the infection in small babies at birth from mothers who are HIV positive. Now, how the disease happens. So that's on this right-hand side scheme where you see in the blue squares is the change in those CD4 cells, the important cells of immune system. When somebody acquires the virus, gets infected, the CD4 cells slowly drop down up to the stage after several years, and this process can take between 3 and 20 years, depending on how good immune response the people might have against the virus. when it comes to the AIDS stage. So the AIDS is only the late stage, the final stage of the disease, the acquired immunodeficiency syndrome. And that's basically hallmarked by the very low CD4 cells and complete non-function of immune system. So these people, when are infected for a long time without the treatment, they basically end up with a whole spectrum of opportunistic infection, fungal diseases, virus-specific cancers, and many other diseases that eventually lead to the exhaustion and death. Now the virus has a sort of independent trajectory. The early acute infection is basically important in seeding the HIV reservoir that I mentioned, and that is relatively short in terms of the weeks, but then the follow-up time for several years when the virus slowly takes over the CD4 cells and increases the amount in the blood, and that's also when people are highly infectious. Now a little bit on the history of HIV. There was an interesting paper published in 1998 from the laboratory of David Ho. That's a brilliant scientist at Columbia University in New York, and we will hear about him in a couple of minutes in the different circumstance. And this is identification of the oldest known HIV isolate that basically has been identified globally. It comes from a Zaire from 1959. It's now Democratic Republic of Congo. And this virus, you see the phylogenetic map. So these are different subtypes, A, B, C, D, and so on, are different subtypes of HIV that are genetically diverse. This virus sits right in the middle. So by genetics, it suggests that it's early precursor of all the subsequent viruses that spread around the world. How long the virus has been in Africa, that really is not well known. What we know so far, it came from the apes or non-human primates through sort of zoonotic transmission, as many other viruses coming from animals to human. But we don't know whether that's been 100 years ago or 60 years ago, probably for quite some time, and it might have spread in the isolated parts of Africa and never actually made it out of the continent globally, until the time of 1981, when in US, in early 1981, several reports published by CDC in their weekly journal, MMWR, described a special symptom that has never been seen before among few gay young people who suffered from this very unusual pneumonia, and as well as a Kaposi sarcoma, which is the certain type of the cancer induced by viruses. And these infections don't occur in people who have normal immunity. So this on its own was very puzzling. Nobody knew where it came from. But what the fact is, by the end of 1981, more than 300 people were diagnosed with this syndrome in United States, and 1/3 of them were dead by the end of that year. So this was the immediate puzzle for the medical community, and it took another 3 years until one issue of Science in the May 1983 published 5 articles giving the clear argument that the HIV virus is actually the cause of the AIDS and those symptoms of immunodeficiency. And you probably know the story of the discussion who actually discovered HIV, whether it were French people or the Americans. The prize went to the French, but I think the Americans did some important work as well. Now, what happened in 80s and early 90s was a huge tragedy. The disease basically spread unchecked for 15 years, and in 1992 it became the #1 cause of the young and middle-aged men death in the United States. By the end of 1995, 500,000 people were infected only in the United States. People didn't know enough about the disease, and what that triggered was not only the medical problem, because there were no effective treatment on prevention, apart from few suboptimal nucleoside analogs that I'll mention in a minute. I think the earliest days of the epidemic of HIV had a huge social impact, not in the United States only, but around the globe, because it was associated with significant discrimination and stigma of people who have been diagnosed with the disease. The people who have been evicted from their housing, people who have been fired from their jobs, people who have been eliminated from their schools just because people didn't know how the virus was transmitted. Science knew, public didn't. And they were very scared. And this brought up very special phenomena that never happened in medical history before and never happened since. And it was a very strong activism that actually came just from the desperation and frustration of not doing enough to understand the disease, to treat the disease, and help people who suffer from the disease as well as their families. So there were demonstrations across the whole United States, Europe, and other parts of the world. The truth is that the movement became quite radicalized, but I think in the end this was the only option how to press the governments into the action. And you can see some of the slogans and the language that the activism and the representatives of the HIV community have used. What is clear now, and many people believe that this all happened, the whole 80s happened under the watch of the President Reagan. And he did too little, too late for the whole epidemic. I mentioned the early treatment and the difficulties. So the first drug was actually approved in 1987, and that's this nucleoside analog called AZT or zidovudine by GlaxoSmithKline. And this was originally anti-cancer drug that was repurposed later on and found to have the activity against HIV. But what you see there is on your left-hand side, the panel is basically change of the viral amount in the blood after the initiation of the treatment with this AZT. And it's clear this is on the logarithmic scale. So you suppress the virus by one log, about tenfold, but within a couple of weeks the virus comes back roaring with full resistance. So one drug couldn't keep the virus at bay, and the resistance emerged very quickly. In early 90s, the analogs or similar compounds from the same class as AZT came to the market, could be combined into two-drug combination. But the only thing it did, it kept the virus a little bit deeper, but the virus came back again. So not even two drugs can hold the virus. And what was the real breakthrough is in mid-90s with the advent of the HIV protease inhibitors, when finally the three drugs combined from two different classes suppressed the virus durably for at least half a year. But even with this regimen of three drugs that became known as highly active antiretroviral therapy or HAART, resistance came later against these different classes. The other problem was with these drugs that they were not well tolerated. AZT had a number of the side effects that were very difficult to tackle, and the same for the protease inhibitors. Just here you see an example of something that's called lipodystrophy, which is basically the syndrome, metabolic syndrome that is triggered by this one particular compound and other compounds in the same class that leads to redistribution of the fat around the body. And this was something that was quite visible to the people who were on these drugs, and that immediately identified them as potential carriers of the HIV, and that only led to more stigma and more discrimination. But it was a true watershed moment, because what it did, the triple combination for the first time after 15 years of the epidemic actually reverted the steady increase of the death due to the HIV and AIDS. And the 1996 and 1997 were the first year when the death rate in the United States started dramatically dropping and ultimately leveled at 1/3 of the peak. The person who stood behind the combination antiretroviral therapy idea was the same David Ho, who described the first HIV isolate. And he was celebrated as a huge visionary because for the first time there was something like a hope. The same phenomena actually happened globally. So you see this curve of the death rate or the death per year that at the peak came up to 2 million. But it shifted to the peak, and the reversion actually was about 8 years later. And that is connected to another singular event. That was the introduction of the President's Emergency Plan for AIDS Relief, or PEPFAR. So we have another president of the United States who, unlike Reagan was successful in international politics. Berlin Wall came down, Iron Curtain came down. but he was really bad about healthcare and HIV. George W. Bush was quite opposite, right? He was not very successful with international politics, but he did a lot for HIV, and he started with the PEPFAR program, which he signed in 2003, and he devoted the defense for 20 years. And this program has been, every 5 years is for Congress up for the renewal. And it's been renewed regularly every 5 years until 2023. And then the differentiation between Democrat and Republican Congress didn't allow the renewal for 5 years. So it's been renewed only for 1 year, so it's up for renewal later this year again. But George W. Bush was the most important advocate of the continuation of PEPFAR. And what it did, over 20 years, PEPFAR actually provided more than $150 billion in funding from the US budget to globally help with the HIV testing, with the distribution of the drugs, with the pricing of the drugs, and that's what drove the curve. The curve is not driven by United States, it's driven by the rest of the world, particularly in Africa. So where is HIV today? It's still pretty serious epidemic. 39 to 40 million people are actually living with HIV, very sadly. More than 1 million of them are children. There is more than 1.3 million new infections every year. And what's also sad is now the whole epidemic shifted to very different population, which are adolescent girls and young women, particularly in Africa and low and middle income countries, which comprise the 60% of new infections every year. There still is a 600,000 deaths every year from HIV and AIDS-related symptoms. But the good news that more than 2/3 or 70% of people actually have access to some kind of the treatment. As I said, the biggest burden for the epidemics comes from Africa, where there are 2/3 of the cases in sub-Saharan Africa. Then 10% in South and North America, 10% in Southeast Asia. Europe is contributing less than 10%. You may be asking now, what is the HIV situation in Czech Republic? And I actually got a little bit interested in this, and I have to thank my colleague Tomáš Cihlář from Gilead, who helped me to provide some of this data, and that's actually from the State Health Institute, or Státní zdravotnický ústav, and I was really impressed by the detail of the statistics that the Státní zdravotnický ústav is keeping since the beginning of the HIV epidemic in Czechoslovakia and in Czech Republic. So you see, cumulatively, there were close to 5,000 cases of the HIV over the course of the 40 years, which contributes a very, very small amount to the overall global epidemics. There are still people dying, and there are more than 250 new infections every year in Czech Republic. When you think about how our neighbors are doing, the rate of the HIV infection is about 3.5 times higher in Germany. It's a little bit higher in Austria and Poland. So we are doing really well in this case, but there are still people who need the treatment, and as I've mentioned briefly in the future, who also need prophylaxis. So let's talk about what actually we have now available in terms of the medical interventions. And there are 3 pillars, basically, when you think how to deal with the disease. It could be treatment, it could be prevention, and ultimately it could be cure. And you see what is actually available. We have many treatments, we have some prevention. So far, we have no cure. That's the state of today. We hope we will do better in future. So let's talk a little bit about the treatment, what the options are, where the drug came from, and so on. Before we get into some details, I just want to provide a little bit bigger context, because I'll be talking about how actually the drugs are being discovered and developed, and so that you sort of have the good awareness of the whole process. The process in principle consists of the 2 different stages. If you want to discover any drug against any disease, you first need to go into discovery and preclinical development stage. And once you get the molecule that you like, that fits the profile that you have in mind, then you move it to the patients and do at least 3 different stages of clinical development of increasing length. But each of these parts can take years and years. Depending on where the preclinical starts, you can start with the target selection, identification, some screening, lead optimization, preclinical testing, and so on. But before it gets into the clinic, it has to pass the gate of the rigorous regulatory review by the regulatory bodies, which in US is FDA, in Europe is European Medicine Agency. There are also other local and regional regulatory bodies that review every step after the drug gets into the people for testing to make sure that people are not harmed, that the drugs that are not active don't go further into the development. And this is one of the pillars of the successful pharmaceutical industry, because if it's not regulated, we would be taking the drugs, half of them would never work, and we will be spending money for it. So this process, the rigor of this process is very, very critical to keep at the level as it is now. And some of you, at least I've seen it firsthand, it got a little bit lenient during the COVID, and we got things like chloroquine and ivermectin and stuff that never worked, and it just didn't pass through the rigorous reviews. Now, this whole process is not easy. It's long, it's expensive, and it's very risky. So you see some numbers here, how long the whole process may work, how much it costs in the hundreds of millions or billions of dollars, and how much failure is there. We don't count much of the failure in the research. Like, we all as a researchers know that a whole bunch of stuff in lab doesn't work, but that's relatively cheap compared to the clinical development. But because of the unexpected toxicity, lack of activity and efficacy in humans, 90% of the drugs that make it through the first gate of the application for clinical testing don't make it through. So it's not easy space to succeed. I have to say the antivirals are a little bit different because we understand the specificity of the viruses very well now. But nevertheless, there were more than 30 HIV drugs that had to go through this rigorous process over the 4 decades. And I just want to walk you quickly through the types of the drugs that are currently available. And it's just the same replication cycle that I show at the beginning. And when we start from the left, when the virus enters the cells, we have a class of the few entry inhibitors. Then we have a class of the reverse transcriptase inhibitors, which are basically 2 flavors, either the nucleoside on so-called non-nucleoside or allosteric reverse transcriptase inhibitors. And the nucleosides or NRTIs were backbone of the antiviral HIV therapy for many, many years. Then more recently, the integrase transfer inhibitors have been actually came to the market, and this is probably currently the most important class of the drugs because it's very efficacious, it acts very quickly, and can be very well tolerated. And finally, during the process of the assembly of the new virus and maturation, the whole series of the HIV protease inhibitors can work. And as of last year, we have another class, the 6th class, which are the completely new HIV capsid inhibitors that so far have the one representative molecule, which is called lenacapavir, and that came from Gilead. So I will spend a little bit time to talk about this later. But before I get there, I think I really have to make the justice to the contribution of the Czech science to the treatment of HIV and other viral diseases that came primarily through the discovery of tenofovir by Antonín Holý, a chemist from IUCB and virologist from Leuven from Belgium, Eric de Klerk, who basically together described and designed the whole class of the new nucleotide analogs that are called nucleoside phosphonates. Now the trick with tenofovir is that it is active against HIV in vitro, but it cannot be given orally because it doesn't have the good permeability across intestinal wall. It's a very good molecule it has to be metabolized inside the cells to the active triphosphate. But that's where Gilead actually came to the picture when in early 1990s, John Martin who was the chief scientific officer at Gilead, actually knew Tony Holley for a long time, and they made a deal, and Gilead licensed the whole package of these nucleotides analogs, including tenofovir. And what Gilead did then, modified them chemically to make them prodrugs. So we have two generations of tenofovir prodrugs. The first one is called TDF, and the other is TAF. But in principle, they do the same thing. They mask the negative charge on the nucleotide to make them more potent and orally bioavailable. And over the years, both TDF and TAF actually became the components of total 14 different pharmaceutical products that have been fully developed and approved for the use either for HIV or for treatment of the hepatitis B. And most of these products are called single tablet regimens. So basically they combine the three. I told you at the beginning, we need at least 3 drugs to keep the virus fully suppressed, keep it from developing resistance and mutating and spreading in the body. So most of these pills are actually, they contain at least 3 drugs. One of them is always the prodrug of tenofovir. So I personally, I don't actually know of any other small molecule pharmaceutical that would end up with so many different pharmaceutical products. And that's really the genuine breakthrough contribution of the Czech science. Now I want to briefly talk about the 2 compounds or 2 pills out of these 14. The first one is called A++, so that contains the first generation tenofovir, and that was the first single tablet regimen that basically packed all 3 drugs into one pill and helped people to make the treatment easier. And the last one, which was approved in 2018, is so far the most successful and mostly prescribed HIV drug that contains the TAF, so the second generation of the tenofovir prodrug, plus the bictegravir, which is currently the best integrase transfer inhibitor. So this is a little bit more visually, what does it mean and how much breakthrough that was for the people who had to be treated with HIV. So in 1996, at the advent of the HIV protease inhibitors, the problem was that these drugs were not stable in the body. They were very quickly metabolized and excreted. So people actually had to take the drugs every 6 to 8 hours, 3 times, 4 times a day. And so they ended up with 30 pills a day, which was very, very difficult, and they had to take it for the rest of their life, otherwise they would die. So the invention of something that can be put together into the single tablet was the major change in the life of the million people, and that was the A++ product in 2006. And then through stepwise improvement, we actually ended up with the product, the Biktarvy, that is one of the most efficacious and most prescribed HIV drugs, as I mentioned. Now, when I talk about efficacy, it's really the almost complete efficacy. So here you see the data from 2 big registrational clinical trials that lasted each of them for 5 years. The first one on the left-hand side is from US. The other one is global, including Europe. And what you see here, the green curve basically captures the proportion of all treated patients who have undetectable virus. So the virus gets completely eliminated from the blood. And that's basically nowadays the standard and the goal of the HIV therapy. And you see that within the less than 1 year, almost 100% is there, and then stay there for the 5 years. Now, the other good news is there is 0 resistance. So with this potent therapy, it's very well tolerated, and people don't have to stop it because the virus doesn't come back. So we have a great drugs now. They still have to be taken mostly once a day. They don't get to everybody. So the other aspect of making the difference in the whole HIV epidemics to actually have the global access to the drugs. And there are mechanisms that go beyond just the drug discovery and development. It's more commercial mechanisms, the mechanisms how the intellectual property is treated, also the mechanisms how the drugs are being and where the drugs are being manufactured for the low cost. So Gilead actually pioneered all of these different aspects and approaches, including the licensing to the generic companies that reduce the cost of goods for the production. contributing to the medicine's patent pool, which is basically the global pool of the patents where every company can take it and under certain circumstances they can start producing the drugs for certain regions. So this brought a huge impact globally. With more than 12 million people actually received the Gilead's HIV medicine in 2020. Nowadays, it's probably closer to 15 million people. And it helped the access to the drugs in more than 120 low and middle income countries. So that brings the question, when we go from here now, we have great drugs. They are well tolerated. They are very safe. They still can be taken once a day. So now we are trying to make something different to make it even easier for because they have to take the drugs for their life, so never forget about that. Make it easier through the concept of so-called long-acting HIV drugs. And what it does simply is visually, within 1 week you take 7 pills, within 1 month you take 30 pills, 3 months 90 pills, 6 months 180 pills, right? And that's still a lot of pills. So there are ways how we can simplify this and put all those 7 pills into 1 pill that can be taken Sunday evening or Monday morning, whenever you think is your best time of the week, or they can be taken once a month, or they can be formulated into injections, intramuscular, subcutaneous, and taken once every 3 or 6 months. Now the trick is, in terms of the drug discovery, it actually needs a very different approach. It needs to have a different types of the pharmaceutical material, drugs that are much more potent, much more stable in the body, so they actually last for that long time and can truly be the long-acting. And there are 2 ways how you go about it. You take the drugs that already exist and you want to modify them such that they gain these properties, or you go somewhere else and think about how to intervene or interfere with the virus replication through new mechanisms that could potentially be more sensitive. And it was now about, how long? 16, 17 years ago when at Gilead, what caught our attention is completely different function of the virus, which is HIV capsid. So unlike the other functions, which are primarily enzymatic, the capsid is a structural feature of the virus. And it's a little container or little box that basically protects the genetic material, it protects the enzyme and brings it into the cell and brings it all the way to the nucleus where it triggers the integration into the host DNA. And this HIV capsid is completely essential. So if it doesn't form, or if it forms in some sort of distorted, bad way, the virus is dead and non-infectious. And it plays the role in many steps along this whole replication cycle, unlike the enzymes that I showed you at the beginning, where there is only one distinct step where they play the role. So we have the many different opportunities as the virus replicates to interfere with its growth inside the infected cells. So just to give you a little bit idea how the capsid actually looks like, it's within the virus, that's this green cone. It's formed from the about 1,500 subunits that are aggregated into these hexamers. The hexamers come together. And this is the polymerization of the protein that occurs at the end of the virus during the maturation. And so ultimately, when the capsid comes together, it looks just like this. So it's cone-shaped structure with these different curves that are caused by introducing the pentamers. Unlike the wall is made of the hexamers, but the curvature is from the individual pentamers. There's exactly 12 of them. And so it's a highly ordered structure where we thought, well, this is a good way how to interfere with the assembly. Maybe if the assembly goes wrong, the virus will be dead. So in 2006, we kind of got this idea, let's start a project on the new inhibitors of HIV capsid. And I have to say what was very important here at this stage was really the contribution of the basic science, because we truly built the whole program on the detailed analysis and understanding of the HIV capsid function and structure. Without that, we would never start that program. And so there are a number of the basic scientists who published over the years on the function and structure of HIV capsid. And we used all the knowledge to build our own assay for the screening of the compounds. We started what's called the high throughput screening or HTS campaign, where we took the whole Gilead library, which was close to 1 million different small molecule compounds, and we tested it for the in vitro polymerization of this HIV capsid. In 2008, we got some molecules that we sort of liked out of it, but they didn't go nowhere by 2010. So that whole direction of 4 years basically gave us some knowledge about the capsid, but we didn't have the right compounds to make a drug out of them. So the second generation lead came in 2010 that looked much better. It was much more tractable. But what was necessary to make about 4,000 compounds one by our medicinal chemists to actually arrive in the end to the final molecule, which originally was called GS6207 and later on named lenacapavir, or Len for short as we call it. And then this became a clinical candidate in 2016. So here you see the 10 years of the work that I basically compressed into the 20 seconds. I don't know how many seconds 10 years would have, probably 2 billion seconds or so. But, and you see how gradually our chemists actually made the improvement of the potency. So on the left-hand side corner, each dot is one compound. So there's more than 4,000 dots on the chart. And you go from the top left-hand side corner where there are almost no active compounds. All of them have a too high concentration to be potent, until you end up at the right bottom where you have a super potent compounds. And the reason lenacapavir is a very potent drug against HIV is actually really interferes with the multiple steps of the replication. As I told you that the capsid has the multiple functions, we actually demonstrated convincingly that it's both the early stage where the capsid enters into the nucleus and disassembles, where lenacapavir blocks that, but it also blocks the assembly and maturation of the process. And that nowadays makes it the most potent HIV drug that basically exists, right? So you see the bar, the size of the bar corresponds to the concentration that is needed to inhibit the virus. So the higher bar, the more drug you need. The shorter bar, the less drug you need. And so the lenacapavir on the right-hand side is 70 times more potent and one of the best protease, HIV protease inhibitors, and many drugs in between. Now, other feature that we tried to build into the drug was actually the stability. And the stability gave us very long presence of the drug in blood. So what you see here is measurement of the blood levels of the drug across 1 year after a single subcutaneous infection, or over the 5 weeks after one oral dose. So lenacapavir is quite versatile. It can be dosed both through mouth as a pill, but also through subcutaneous injection. And we've worked out the amount of the drug that's needed to have enough of the drug floating in the blood to continue the inhibition of the virus, and has very long half-life from both routes of administration. And so with this knowledge, we initiated the clinical development in 2016, or actually 2018 here. I'm sorry, because there was about a year of the preclinical development through these different phases. And in the end, we obtained the approval in the end of 2022 as a product called Sanlenka. And this is the first HIV capsid inhibitor and the first drug that can be given only once every 6 months, unlike the pill every day. Now, we built a whole portfolio of the HIV long-acting drugs based on the lenacapavir. The problem here is if you want to use lenacapavir for treatment, you need at least one partner drug. And in order to leverage the potential, you need a drug that can be given at the same frequency. So we have a number of the programs that now are focused on finding the integrase transfer inhibitor, so the different complementary class that potentially can be given once every 6 months as well, and that would allow for the complete combination regimen. We have the oral programs both for weekly and monthly administration now, and several combinations that actually are in the later stage of the clinical development. One of them I would like to mention here is this is the combination of lenacapavir with neutralizing antibodies. So it's one small molecule and one biologics or two biologics. And this is the only currently the full regimen for HIV treatment that can be given once every half a year that is in the later stage of clinical development. But where we think is the real potential of lenacapavir is the actual prophylaxis. And so let's talk a little bit about what the prevention or the prophylaxis is. Vaccine comes to mind as the best prophylaxis, right? We all are vaccinated against many infectious diseases. Almost 30 years of the HIV vaccine research yielded 0 positive result. The subunit vaccine, viral vector-based vaccines or their combinations don't work. They generate the antibodies, but the antibodies don't neutralize the virus. They don't block the virus. And why is that? That's because the HIV is genetically very, very diverse. And particularly the surface of the virus that is recognized by antibodies is very diverse. And so when you look at diversity of influenza, for example, in 1 year versus diversity of HIV just in one country in the similar year, you kind of come to the conclusion of comparison of the small baby tricycle to the big moving truck of being flu and HIV. And so, you know, we have difficulties every year to develop the match the flu vaccine. Now for HIV vaccine, this is 1 order of magnitude bigger problem. But what is good news, there is alternative solution. And that solution actually comes from using the antiviral drugs themselves for prevention. So not just for treatment, but also for prevention. But there are 2 ways how you can go about it. One is when people who are infected are on the suppressive antiviral therapy. So if they don't have the virus in the blood, they cannot transmit the virus. And that's a part of the prevention. They cannot transmit the virus to their partners. And so in 2016, this huge global movement came up, which is called U equals U, which stands for undetectable equals untransmissible. And this is based on the clear scientific data. If you don't have the virus in the blood, you cannot infect anybody, period. That's how it works. And so a lot of people came and responded to this message, particularly across the HIV community. And that's why it's important to keep so many people on the suppressive treatment who have the virus in the blood. So that's one way how you go about it. The other is you protect the people who are healthy, not infected, but are at higher risk of acquiring the virus. And that's so-called pre-exposure prophylaxis. So there are a couple principles that can be used for that and different ways how you go about it. Oral single tablet regimens, once daily taken, either Truvada or Descovy. So you see they contain the tenofovir prodrug. So these are the Gilead products. And Truvada was the first prophylactic product approved in 2012, actually. So that's on the market for more than 10 years. There could be the vaginal ring. There is one that is currently available in some countries in Africa. That was developed by one of the non-governmental NGOs. And it contains the non-nucleoside reverse transcriptase inhibitors that get slowly released, but that protects only women. Now we also have the most recent product, which is the integrase transfer inhibitor that can be given by injection every 2 months. So there are ways how you can actually decide what works best for you. Now, what is also very important to recognize that if people don't take the PrEP or the pre-exposure prophylaxis, they are not protected. So there is a very strong movement to actually expand the use of the PrEP, especially in the countries where there is a higher frequency of the HIV occurrence. And so this program started in 2016 of the introduction of the global PrEP. And, you know, it started with 100,000 people, mostly in sub-Saharan Africa. And over the years, it grew, and now it's growing by about 50% every year. And we have more than close to 7 million of the people globally on the pre-exposure prophylaxis. Now, the trick of the pre-exposure prophylaxis is it works if it's taken. So it's not easy to take if people feel healthy. Many of them don't feel compelled to take the pill every day. And that's, I think, where the long-acting antiviral drugs can actually can come in a picture and make it much easier to take the prophylaxis. So Gilead, a few years ago, started very ambitious clinical development program to test the lenacapavir for the pre-exposure prophylaxis in the countries where there is high incidence of HIV, both in sub-Saharan Africa and in Americas and Southeast Asia. And we had to come up with the innovative drug designs because the oral pills have been already approved before for the prophylaxis. So they represent what's called a standard of care. Now, if there is a standard of care, people can compare in clinical trials a new drug against the standard of care, but not against the placebo. And now, if the standard of care is very, very active, it's very difficult for the new drug to actually show the difference. So we have to come up with a completely innovative design of these clinical trials. And so it's more than 8,000 people of the different populations that are being tested. These 2 trials are fully enrolled, and we expect the results to learn by the beginning of the next year. And if it works, it would be once every 6-month injection. So I think that's going to be a really major breakthrough for the addressing the whole epidemic of HIV. So here I would like to really acknowledge the Gilead team of many hundreds of the people and several of the external collaborators, primarily from the academic field, who help us to set up the whole program on lenacapavir. And you see these different functions that all need to come together, basically at different stages of the preclinical and clinical development to make a drug that can be given to the patients. And it's really teamwork, and otherwise it doesn't work. So let me talk a little bit about the final part, which is the HIV cure. And the not so good news is we don't really have the HIV cure. And the reason for that is because of that latent HIV reservoir that is established relatively early during the acute infection. So there are certain cells in the body that when they get infected by HIV, the CD4 cells, they don't die. They somehow are able to integrate the virus into their DNA, and they survive the attack of the virus, and they are sort of the carriers of the viral copies. And when the cells get activated, they start shedding the virus. And then if you stop the therapy, the virus spreads again, so people never get cured. And this is this stable reservoir. Then when people are on the antiviral drugs, the reservoir is suppressed, so there is no production, no expression of the virus. The virus is invisible to the immune system, and the antiviral drugs cannot get to it, so it doesn't eliminate. So we need completely different approach how we can potentially activate the latent reservoir in the presence of antiviral drugs so that virus doesn't spread. We try to reduce it and maybe eradicate the reservoir. And this can happen with somewhat different approaches, which are primarily based on sort of immune therapy. So some of you may know the immune therapy against the cancer. So similar principles can actually be applied to attempt the HIV cure. And what the cure really is, if people start, stop the antiviral drugs, the virus doesn't come back, right? That's the cure, basically. And so there is more than 25 years of the history of the idea of the curing HIV. And it started with describing and understanding this latent reservoir. And there are a few patients who actually have been cured of HIV, but they are really handful. And they all have been cured through this very risky, difficult procedure, which is the hematopoietic stem cell transplantation. So these patients, whether it was the Tim Brown or Ram Castillo, they all had the hematological cancers. They needed the hematopoietic stem cell transplantation, and they got the cells from the donors who have been actually genetically resistant to HIV. So they can repopulate their own immune system that became resistant to the virus. So the virus were nowhere to go, and they were ultimately cured. But this is the process that cannot be applied to millions of the people. So there are now actually approaches and the directions that are being translated from some preclinical testing into the clinical studies as we speak now. So we have a treatment, we have prevention, and maybe we have cure eventually in future. But that's not enough to end the HIV epidemic, right? This all has to be under the roof of the access to the drugs, the equity health around the whole world, the engagement with the local communities to understand the risk of the HIV, how to introduce the prophylaxis into the communities, and the global partnership that can end the HIV epidemic, ideally for everyone everywhere. And only then and then we can eliminate the virus from the surface of the earth, and we are still far from it. So let me give you into the final addendum here, which I kind of couldn't resist to compare the HIV pandemic and the more recent COVID pandemic. And these are completely different viruses, of course. Just the way how HIV is a silent killer. It takes the years until it kills somebody who is infected. COVID-19 can kill person in 3 days. Now the difference also is the whole time course of when the virus was identified, where the first drug was available. We don't have the vaccine for HIV. We have all that for COVID-19, right? And one can say, well, maybe this was because the virus was such a big problem globally and everybody had to deal with it quickly, unlike the HIV. I mean, the start on the HIV epidemic was very slow, as I told you at the beginning. But the HIV taught us a lot, actually, that we could then use and leverage in our response to COVID-19. And I just showed a few examples of the molecular diagnostics, the PCR, that has been developed largely to HIV to deal with diversity of the virus. Without that, all the COVID variants would be much more difficult to actually detect, identify. The principles of the antiviral drug designs, many of the drug classes that have been developed for HIV, the very similar mechanisms have been directly translated to COVID-19. The mRNA technology. Not everybody is aware that Moderna started on the mRNA delivery into HIV people a long time before they succeeded with the COVID-19 vaccine. The infrastructure of the clinical trials. Many clinical trials in COVID-19 have been actually conducted and executed by people who work in the clinical trials on HIV. The expansion of the manufacturing processes, the drug access models, and the partnerships and collaborations around the world. So let me just give one example that I couldn't resist, and that is the remdesivir. Remdesivir life started, the history is not that short, right? The approval was very quick for COVID-19. But the story of remdesivir started in 2019 with this precursor, which is the nucleoside analog. So it's from the same class as tenofovir on the AZT, the first HIV drug. It just uses the different chemistry and different structure. It was originally started at Gilead as the program for hepatitis C, but they didn't lead anywhere because we found the better molecules to address the hepatitis C epidemic. But it turned out that this precursor molecule has the activity against many, many different viruses. So we then switched to a respiratory virus called RSV, or respiratory syncytial virus, and designed a prodrug of that nucleoside. And the chemistry that is there has been used 20 years before for the HIV drugs. It's the very same principle that allowed the design of the remdesivir. And over the years, in the partnerships with many other collaborators, we have actually tested remdesivir against many, many different viruses, including the existing coronaviruses. So in 2020, when the pandemic started, we actually were ready. We had more than 5,000 treatment regimens for clinical trials. We have safety in 400 people, and we have very strong preclinical data to show that the remdesivir would be active against coronaviruses. And within less than a year, through the collaboration with many clinical investigators, we were able to collect the data that led to the approval of remdesivir as the first antiviral drug for COVID-19. And I also want to mention that all the principles of global drug access that we have built and pioneered for HIV have been successfully used to make the remdesivir available globally to more than 14 million patients, and at least 8 million of them in the low and middle income countries. And finally, the partnerships. Remdesivir, lenacapavir was a different program. We were really focused on the internal collaboration of the different functions to bring the molecule into the clinical development. Remdesivir was the very opposite story. We worked with many partners who actually understood all these different viruses and coronaviruses because we didn't have the expertise at Gilead. And through that, we were able actually to quickly prepare the remdesivir for the approval as the first antiviral drug. So with that, I would say děkuji vám za pozornost. Doufám, že to bylo inspirující a velice rád odpovím všechny vaše otázky.
Děkuji za skvělou přednášku. A nyní je čas na otázky. Dotazy, prosím. Tak, já vám ji přinesu.
Já bych vám šel naproti, ale nemám možnost úplně.
To dáte, dobrý.
Děkuju. Loďánek, jestli vás to zajímá. Vy jste v úvodní asi třetině nebo čtvrtině té přednášky se věnoval historii HIV a ani jednou, jestli si dobře vzpomínám, tak jste nezmínil spojitost jako s geji. U níž mluvil se tam o tom, že HIV bylo jako nějakou primárním důvodem smrti mladých mužů a většinou to byli mladí gejové. A zmínil jste demonstrace proti diskriminaci lidem s HIV, což většinou byli jako LGBT lidi s HIV, kteří protestovali nejenom za to, aby se nediskriminovali lidi s HIV, ale i za to, aby se nediskriminovala ta komunita. A kdyby nebyla ta frustrace z těch jako násobných úmrtí v rámci té pandemie, tak by jako pravděpodobně nedošlo ani ke Stonewallu a k těm protestům o zrovnoprávnění jako neheterosexuálních lidí. A mě by zajímalo, z jakého důvodu jste tady tu integrální součást historie HIV vůbec nezmínil.
No, to se omlouvám, jestli vám to uteklo. Já jsem měl s váma naprosto souhlasím. První, jako to je můj první bod, který bych já rád poznamenal. A naprosto máte pravdu, že to bylo LGBT hnutí, který jako v závěru vyústilo v to, že i vlády Spojených států a všech dalších zemích přiznali řadu opatření a uznali vlastně tu pozici. A myslím, že je dobré to říct, že všechny ty demonstrace nebo naprostá většina byla organizována a iniciovaná tou LGBT community. So, jestli jsem to neřekl jako zcela zřetelně, tak se za to omlouvám, je to naprostá pravda.
Děkuji moc. Další otázky, prosím.
Děkuji za moc pěknou přednášku. Mě by zajímalo u Lena Kapaviru, jak je to s rozvojem rezistencí, jestli u vlastně ten mode of action je vlastně cílení jako strukturního proteinu, jestli tam vznikají rezistence a do jaké míry je pak potřeba kombinovat s dalšími látkami.
To je dobrá otázka, vznikají a v podstatě na jakoukoliv látku se dá vyvinout rezistence. Myslím, že když se podíváte na celou tu škálu těch HIV inhibitorů, tak integrázové inhibitory mají to, čemu se říká jako nejvyšší bariéra k rezistenci. A zhruba nejnižší mají ty nenukleosidové alosterické inhibitory reverzní transkriptázy. Lenacapavir někde mezi tím. A ta rezistence byla dokumentovaná v klinických zkouškách. A ten hlavní důvod k tomu byl, že vlastně ty zkoušky a ta indikace, která je teď povolená s lenacapavirem, je vlastně léčba pacientů, kteří momentálně, protože nemáme žádnou další látku, kterou bychom s tím spárovali v té stejné frekvenci vlastně toho dávkování, tak jako add-on terapii nebo jako další přídavek k terapii je vlastně povolená pro pacienty, kteří mají rezistenci na několik typů HIV inhibitorů. A tam, když přidáte tu jednu látku, tak v podstatě je to taková jakoby monoterapie. Takže v několika pacientech se ukázalo, že ta rezistence vlastně se vyvinula, ale to bylo proto, že oni neměli žádnou další látku, která by byla aktivní, takže ten lenacapavir to držel nějakou dobu a potom zřejmě, řekněme třeba po půl roce se objevily rezistence. Když to zkombinujete s další aktivní látkou, tak se tam ta rezistence nevyskytuje zdaleka tolik.
Děkuju. Já mám asi 15 otázek, ale nechci zneužívat z toho mikrofonu. Já bych se vrátil k remdesiviru. Já vím, že covid už je za námi a nikdo o něm nechce mluvit a vzpomínat na něj. Přesto, jestli tomu správně rozumím, nevýhoda remdesiviru bylo, že se nedal používat orálně, takže se dával až v nemocnicích, což bývalo často pozdě. Když tak mě oprav, jestli to říkám zjednodušeně. Pracujete vy nebo Gilead na nových formách podávání remdesiviru nebo jiného protivirového léku proti covidu?
No to už v podstatě v těch začátcích covidové pandemie vlastně napadlo, že to je ta hlavní limitace remdesiviru. Že jo? Takže na to, aby se dal vlastně používat na to, aby zabránil hospitalizaci, což jako je ten ideální scénář, tak na to opravdu potřebujete orální látku. Takže my jsme vlastně použili ten původní nukleosid, který jsem tam ukazoval úplně na kraji toho slajdu z toho roku 2009 a snažili jsme se ho modifikovat tak, aby jako byl orálně dostupný, což se nám asi po roce nakonec podařilo a na konci někdy zhruba v polovině roku 2021 jsme měli první kandidát, který skončil v těch původních klinických zkouškách vlastně do konce roku 2021 a teď dokončujeme 2 velké klinické studie ve fázi 3, kde teda ten velký problém je, že vlastně dnes, když chcete měřit účinek látky na to, aby zabránil hospitalizaci, tak v první řadě se ta hospitalizace musí vyskytnout v té klinické zkoušce. Takže ten náš problém byl, že ty klinické zkoušky, které jsme dodělali, tak jsme tam neměli dost hospitalizací, protože lidé už prodělali několik covidových infekcí a většina z nich byla vakcinována, takže ta nemoc byla velice mírná, takže jsme nemohli vlastně detekovat ten rozdíl, který by přinesl ten treatment s tou orální látkou, ale máme teďko nedávné jako velice nadějné výsledky na opičích modelech eboli a různých filovirů, kdy vlastně ta orální profylaxe zabrání tomu, aby se ten virus eboli rozšířil v primátech a to je vlastně ta nová fáze, kam bychom tu látku chtěli tlačit, protože žádná orální jako profylaxe na ebolu a filoviry konkrétně v této chvíli neexistuje.
Děkuji moc. Koukám se kolem. Prosím.
Dobrý večer, děkujeme moc za krásnou přednášku. Já navážu také vlastně na tu indikaci u dalších RNA virů, jestli se uvažuje o RS virech jako target další pro remdesivir, nebo tu jeho perorální formu.
RS myslíte respirační syncytiální virus? No, pro tu orální formu, ano, nad tím přemýšlím, máme předklinické zkoušky teda hotové už a tam na různých modelech je to aktivní, takže teď se jako řešíme tu otázku toho, jak to nejlépe dostat do kliniky právě na to RSV.
Dobrý večer, děkuji za tu přednášku. Já jsem se chtěl zeptat na něco možná trochu stranou vědy jako takové. Jste špičkový vědec, dostal jste se do vysoké manažerské pozice v rámci originální farmaceutické společnosti. Je něco, co vás jako vědce překvapilo na tomhle manažerském postu? Je něco, co třeba bylo kontraintuitivního, co jste nečekal?
Překvapilo mě to, že máte hrozně málo času na vědu a že ta manažerská práce pak už není moc o té vědě, ale je úplně o něčem jiným. A myslím, že to jsou hrozně důležité posty, protože tam vlastně kloubíte to, že rozumíte té vědě, ale zároveň to musíte dělat s těmi týmy těch lidí. A to jste viděl tam na tom seznamu, to jsou lidi z různých oblastí, s různou specializací, mluví anglicky, ale v podstatě je to různá terminologie a my je nějak musíme dát dohromady, aby stáhli za jeden provaz a posunovali se stejným směrem. Takže to pro mě vlastně bylo to probuzení, že člověk může být skvělý vědec, ale když není schopen nějakým způsobem dát lidi dohromady, tak v podstatě z toho nic moc nebude, aspoň v té branži, v které já se pohybuji.
Děkuju. Prosím.
Děkuji za přednášku. Možná bude mít trošku kontroverznější otázku. Je ten lék nebo nějaký z těch léků, co vyrábíte proti HIV, dostupný v Rusku aktuálně?
No, to asi nejsem nejlepší člověk, který by na to dokázal odpovědět, ale myslím si, že ano. Já nevím, jak vlastně zvládáme, protože já jsem z oblasti vlastně toho výzkumu a research, takže já přesně se musím přiznat, že nevím, jak se ta komercializace jak se k ní přistupuje v Rusku, ale je jasné, že HIV v Rusku je čím dál tím větší problém. Takže určitě by se měly najít cesty, jak by se tam ty nejlepší látky mohly dostat.
Jenom pokud já vím, aspoň z počátku to bylo tak, že léky proti smrtelným chorobám nepodléhaly blokádě, takže že bylo možnost s nimi obchodovat. Ale taky si přiznám, že současný stav neznám. Další otázka, já jich mám spoustu. Kdyby tady byl kolega Machala z Bulovky, tak bych se tě zeptal na takový ten projekt 90-90-90, který, o kterém se často mluví. To znamená, bylo by ideální chorobu vyléčit, to zatím neumíme, tak matematické modely naznačují, že pokud se podaří diagnostikovat 90% všech lidí, kteří mají virus, což není jednoduché, u 90% z nich léčit účinně a u 90% z těch léčených dostat tu virémii na nulu nebo na nedekovatelnou hranici, tak vlastně efektivně zabráníme šíření viru, protože potom bude těch nemocných schopných šířit chorobu tak málo, že vlastně už nebudou schopni šířit efektivně chorobu. Je tohle něco, o čem přemýšlíte a co by mohlo být cílem a vlastně způsobem, jak ten virus zastavit?
No, já myslím, že to je reálná možnost a určitě bychom na to nestačili sami jako Gilead, ale tohle je kampaň Světové zdravotnické organizace, která začala tím 90-90-90 a i když to se nikdy nedosáhlo globálně a paradoxně vlastně nejúspěšnější v tomhletom principu byly některé africké státy jako Botswana, Uganda, myslím, kde se blížily nejvíce tomu 90-90-90 a když už bylo více států, kteří se k tomu principu přibližovali, tak Světová zdravotnická organizace řekla, tak to uděláme 95-95-95. Takže teď ten cíl není 90, ale je to 3x95 už. A ten ideální stav je dosáhnout to do roku 2030. Jestli to bude možné nebo ne, to uvidíme, ale myslím, že k tomu by mohly přispět částečně aspoň ty látky s tímhle long-acting potenciálem, kdy bude jednodušší je vlastně přinést i do těch oblastí, kde je horší distribuovat ty orální látky.
Děkuju, koukám kolem sebe. Tak já položím poslední otázku. Jaký virus působí příští pandemii?
Respirační. Ale nevím, ptačí chřipka, je to reálná, podle mě to je reálná šance. A ještě se asi toho musí dost stát, aby ta ptačí chřipka, aby člověk byl jako opravdu dobrý hostitel, ale jako ta infekce skotu ve Spojených státech asi překvapila každého. Že jo? A ta škála vlastně těch zvířecích druhů, které jsou hostitelem pro ptačí chřipku, se rozšiřuje a tím vlastně se zvyšuje šance, že ten virus se bude modifikovat tak, že eventuálně bude schopen přeskočit i na člověka. Takže myslím, že bychom neměli váhat a měli bychom se zaměřit víc na chřipku.
A máte aktivní program vývoje virusu proti chřipce?
V téhle chvíli skoro začínáme s některými, ale ještě nemáme. Teda, no.
Super, děkuji. Dámy a pánové, já myslím, že se mnou budete souhlasit, že to byl fascinující večer a nádherná přednáška. Prosím, připojte se ke mně s poděkováním Tomáši Cihlářovi za doskvělou přednášku.
Děkuji.
A teď bych vás rád ještě jménem zprávy